Hello and good evening. Welcome to tonight's webinar. I see we've still got some people filtering in, so we'll wait just a moment and then we will get started. Okay. Well, thank you for being here this evening. My name is Jessie Jones and I am the volunteer coordinator for Coast Watch, the Coast Watch program of the Oregon Shores Conservation Coalition. And Oregon Shores is a regional group that works to protect the environment of the entire coastal region from the Columbia River to the California border and from the crest of the coastal mountains to the edge of the continental shelf. In addition to organizing Coast Watch and citizen science, Oregon Shores tackles a wide range of conservation concerns, including land use planning, water quality, endangered species, shoreline management, and marine conservation. Through Oregon Shores Coast Watch program, volunteers adopt 1 mile segments of the Oregon shoreline. Coast Watchers keep watch for a wide range of natural phenomena and human impacts. And Coast Watch also organizes a number of citizen science projects. New volunteers are always needed. So if you are not now a Coast Watcher and you would like to be, you can please contact me, and my information, my contact information will be at the end of the program. So now I would like to welcome our very special guest, McKenna Hainey. McKenna Haney was a marine science educator at the Charleston Marine Learning Center and has a master's degree from the University of Oregon's Oregon Institute of Marine Biology, based in the very special Charleston, Oregon on the South Oregon coast. Her research there focused on the morphology and behavior of the basket star and western spiny brittle star. She will be starting her PhD at Oregon State University this summer, investigating how changing environmental conditions play a role in marine invertebrates, particularly in marine invertebrate diseases, excuse me, and particularly the sea star wasting disease, which she'll talk about during tonight's program. And we'll have to have her back as she will continue to do some great research and have great information. McKenna, it's so great to have you here. It's nice to see you. I first met McKenna at the Charleston Marine Life Center in 2019 or 20. So wonderful to have you. And take it away. Thank you for being here, everyone.
I'm super excited to talk to you all tonight. So thank you so much for being here. And my goal is at the end of this presentation, you all will admire brittle stars, Ophiuroids really as much as I do. So with that, the title of my talk this evening is Fantastic Brittle Stars. Honestly, it will be over Ophiuroids, not just brittle stars, and then where you can find some of them. So let's get started. So what are brittle stars? When I was working at the Charleston Marine Life Center, I would get this question a lot. And brittle stars belong to a class of echinoderms called Ophiuroids. And Ophiuroids are composed of more than just brittle stars. They also have basket stars and serpent stars in them. And I always get asked the question, are they sea stars or starfish? They are not. While starfish and sea stars are also a member of the Echinodermata phylum, they are not the same as Ophiuroids. So in Echinodermata, you have your feather stars, your sea stars, your Ophiuroids, your sea urchins, and your sea cucumbers. So they are all related, and brittle stars are an important part of that phylum. So the word Ophiuroid has a Greek origin from the word ophis, meaning serpent or serpent-like. And it's very fitting because these animals have very serpentine-like arms. Brittle stars tend to have shorter arms, and serpent stars, of course, as their name describes, have very serpentine-like super long arms that help them wrap around corals and hang on in currents. And then basket stars also have 5 arms, but they branch, they bifurcate, and when they feed, they form a basket shape, which is where they get their name from. So these are the 3 main groups of this Ophiuroid class. So what makes an Ophiuroid an echinoderm? In order to qualify as an echinoderm, an animal has to meet several key characteristics. And those characteristics are pentaradial symmetry as an adult, bilateral symmetry as a larva or an earlier life stage. They have a water vascular system, a calcareous endoskeleton system, this special tissue type called mutable collagenous tissue, and they're also deuterostomes. So now let's go through these one by one in a little bit more detail. So pentaradial symmetry as an adult means basically that you can just divide the adults up into 5 equal parts. And their larvae, or the earlier life stage of these individuals, have 2 equal divisions. So they are bilaterally symmetrical. So you could cut them in half or fold them in half, and you'd have 2 equal parts. So the water vascular system is this really unique, I kind of like to think of it as a piping system with special capabilities that help these organisms do the things they do, like move oxygen molecules around their body, move carbon dioxide molecules as well. And this piping system goes into each and every tube foot down each arm. And this is important because it is able to pressurize these tube feet, and then the muscles in the tube feet can use this pressurized system to do work off of. So they're able to move their tube feet based on pressure, and it helps them locomote and find food and exchange oxygen and waste molecules. So the water vascular system is really important to this group of organisms. And they're also deuterostomes. So deuterostomes is a fancy way of saying the mouth forms second to the anus during the animal's embryological development. We are also deuterostomes. Most chordates are. So echinoderms also have a calcareous endoskeleton. And while they are true invertebrates, they don't have bones. They've adapted these hard bony parts out of the shell-like material, which is mostly calcium carbonate. Now, Ophiuroids have this really cool meshwork of this calcareous material that their cells build to be porous and lightweight, but very, very strong. So this really cool pattern that their cells build is called stereome, and it helps them support their body systems. It helps their muscles do work, kind of like our skeletons support the muscular work that we do. So it's a really cool way to navigate not having bones. Now, all echinoderms also have a specialized tissue type called mutable collagenous tissue, or MCT for short. And this special tissue allows for rapid arm dropping. And this rapid arm dropping behavior is probably where brittle stars got their name. This behavior is a predator evasion tactic. So imagine a crab comes up to a brittle star and grabs hold of an arm with the intent to eat it. Well, that brittle star can drop the arm very quickly that crab has grabbed a hold of, and it can move the rest of its body and the rest of its legs away from that predator that only gained the arm. So it's this adaptive predator evasion tactic that has served them well for a very long time. So long, in fact, that Ophiuroids are very prevalent in the fossil record. Now, I really love Ophiuroids, and I'm fascinated with them because every time I look at them, I know I'm looking at a living fossil. So the modern Ophiuroid ancestor emerged somewhere during the early Ordovician period, which is about 500 million years ago. It's just crazy to me that, you know, we look at sharks and we think they're amazing living fossils. We look at alligators and crocodiles and think they're also amazing living fossils. But brittle stars predate both of these, and they are so old that they're often found in fossil assemblages with trilobites. You guys can see there's a photo here up on the screen of a beautiful fossil assemblage with lots of brittle stars and trilobites in it. So they've been around on Earth for a long time. And today there are about 1,900 extant species all over the globe. I also get asked, how do they eat? And since they've been on Earth a long time, they've had this long time to figure out different modes of eating. So one way they can eat is deposit feeding, which is basically they crawl around on the rocks and substrate and gather detritus and little bits of food that have settled out on that rock. And when they gather the food, they do so with their tube feet, which are often covered in a sticky mucus. And they'll wrap the food particles up in this mucus until it makes like a little ball shape. And then they'll pass that ball of food and mucus down the arm to the mouth, which is on the underside, and then eat it that way. Many Ophiuroids are also suspension feeders. These include animals like basket stars, you can see here on the screen, and other brittle stars like the Ophiothrisids, which is down the corner, the pink brittle stars there. And I get often asked, how do they catch planktonic prey? Well, they stick their arms up in the current, and sometimes their arms are covered in like little spikes and spines, and then they use other arms to hold on to their substrate. And they basically wait for ocean currents to bring plankton to them. But there are some planktonic prey that are larger, like larval fish and krill, and things that have hard carapaces that might be hard to hold on to. So when I was at the research station at OIMB looking at basket stars, I took some images of their arms close up and under scanning electron microscope, and I found these amazing hooks, and these hooks that not only covered the entire arm length, but each hook moves on its own individual joint, and it has its own set of muscles. And I was looking at these images and I thought, I have seen this structure somewhere before, something very similar. And it finally dawned upon me that I've seen this before in cat claw diagrams. So you can see here, there's a picture of a basket star arm hooks on the right, and then a cat claw diagram on the left. And you can see they're very similar in structure, but also where the muscles attach to them. And that function is almost identical. It's almost exactly the same. But this structure and their ability to rotate on that joint allows them to catch large prey, especially with hard carapaces, and then hang on to them. Something else that is also super cool is that their tube feet secrete paralyzing mucus. So not only do the prey get stabbed with these little hooks, but they get covered in paralyzing mucus before being delivered to the mouth and eaten. I find their different modes of feeding incredibly fascinating. There are some species that are a little bit more predatory than the suspension feeders, and these are the sit and wait predators or scavengers. There's a species of brittle star called the giant green star. I think it's native to waters around Australia. But it has this really creative feeding tactic where it lifts its body up off the substrate and it creates this little dark cranny. And this little space is very appealing for small reef fish looking for a place to sleep at night, where they, won't be eaten by sharks or other nocturnal predators. Well, for this poor unsuspecting reef fish that happens to use this space as a potential shelter, once night falls and the fish falls asleep, the giant green brittle star will slowly twist itself, closing the gaps between those arms, trapping that fish that thought it was gonna have a nice place to sleep. Then it eats it alive. So it's incredibly amazing. Such a weird and unique predation tactic. And then there are other more opportunistic predators, like these brittle stars displaying a mobbing behavior on this squid. So there's an incredible video on YouTube where I've dropped the link below on the screen of a submarine. It's taking video of the seafloor and it's looking at these brittle stars. And this unsuspecting squid just barely touches this brittle star arm. And of course, the brittle star instantly grabs the squid, pulls it down, but that movement attracts its neighbor brittle stars. And they also join in on taking down this squid. So this squid had a very bad day. It was met with by a mob of brittle stars that subsequently devoured it. If you're interested, you can find that video on YouTube with that link, or just by Googling brittle stars attack and eat squid. It should be the first thing that comes up. I highly recommend it. It's an amazing video. So reproduction. Brittle stars have several different modes of reproduction, but most are gonochoristic, which is a fancy way of saying they have separate sexes. So individuals will either be males or females. However, there are plenty representation species that are hermaphroditic. Most are free spawning, which means they will release eggs or sperm into the water and just hope that they meet in the water column and fertilize while in the plankton. However, some females do retain eggs inside their body in special structures called bursae, and they have internal fertilization. And they will brood their young, which means the embryos will develop inside those bursae. They'll hatch out into little tiny versions of the adults, and then the small juveniles will crawl away. And then the last type of reproduction that some species can do is called physiparity. And physiparity means division through fission. So imagine you have a species of brittle star that can do this and you cut it in half. That's your fission. But then the two halves will regrow their missing parts and you'll have two new adult brittle stars that have regrown all their parts, and they'll share the same DNA. So they'll essentially be clones of one another, which is super cool. So most species have a planktonic larva. The Ophiuroid larva is called an Ophiopluteus. Different echinoderms have different types of pluteus, but you can look at a pluteus and figure out if it belongs to an Ophiuroid by looking at the length of the larval arms. And the larval arms are these little structures that look like antennae sticking out of this spaceship looking creature. So they have these iconic long larval arms. However, a few are direct developers, which means the egg and embryo will develop into this kind of blob-like looking creature that will eventually begin to show that penta-radial symmetry and crawl away as the small version of the adult. For the pluteus forms, the juvenile body will begin to develop on the larval body, and the larval body will begin to kind of shrink up as the juvenile body grows. You can kind of see that happening on this picture in the bottom right-hand corner. This particular specimen, the juvenile body is probably about to drop off, and that juvenile will fall out of the plankton and begin its life as a miniature version of the adult on the sea floor. So I wanted to include this study in my talk because I thought it was super fascinating and a really cool use of modern tools to look at ocean animals. And this study was done by Lanshoff and Griffiths in 2015, and they looked at brooding species of brittle stars by using a combination of ultrasound and X-rays. And they were able to produce these amazing 3D images that show exactly where these juveniles are within the adult body. And it showed that the juveniles develop within the bursae. And again, these bursae are kind of like little pockets that you can find on either side of the arms of these animals. And I'm going to talk about the bursae in particular, because one, they're a very important structure for these organisms, but two, they're also the main focus of my research, which I'll talk about in just a moment. But functions for these bursae are oxygen exchange, waste removal, so like carbon dioxide molecules, as well as juvenile brooding, and gamete expulsion. So sperm and eggs leave the body through the bursae. So most of my research happened at the University of Oregon's Institute of Marine Biology in Charleston, Oregon. I was also super lucky enough to join the Geo Study Abroad program and do some of my research at the Smithsonian Tropical Research Station in Bocas, Panama, on a related species. So I was really lucky to have representatives from cold temperate water, warm temperate water, and warm tropical water. And I'll talk about why those differences are important in just a moment. But I was introduced to basket stars as an undergrad at OIMB, and I was totally fascinated with them. So my research journey started with describing how they feed and how they catch plankton and then move their arm to the underside of their body and deposit the food into their mouth. And while I was watching them eat, I noticed this really awesome behavior and this movement of their body disc. You can see, hopefully see in this video, of their body disc pumping up and down. And I didn't really know what that was, but I was curious about learning more. So I went to the literature and tried to find any information that I could on it. And what I did find took me all the way back to records from the early 1800s, where this movement was described as bursal ventilation and that it probably had respiratory functions, but that was it. And then nothing was confirmed or denied after that. So I decided that I'm interested in this research question. I'm going to set out to find evidence that either supports or denies that this behavior is respiratory in nature. So I started by looking at what happens when you add food to an aquarium with a basket star in it. So my thought process here was, in general, animals have to increase their respiration rate when feeding, when actively feeding, and metabolism goes up. So if I'm going to see any behavior change, I would likely see that with the introduction of food. So one by one, I added 9 specimens to a test aquarium and added this krill smoothie, which was basically blended krill and seawater. And then recorded the rate that this body did this bursal ventilation behavior. And every single individual that I looked at increased this rate of bursal ventilation during feeding. And the statistics that I did on this data suggested that yes, this bursal ventilation does significantly increase when actively feeding. And that brought me to the end of my bachelor's degree. I finished my honors thesis working on that project, and I still had questions and was lucky enough to start a master's program in the same lab, working on this question to flesh it out a bit more. So what I did next was lower the oxygen levels. And the thought process behind this was when you lower the oxygen levels, either in the air for us air-breathing mammals or in the water for aquatic animals, you're going to have to work harder to gain the oxygen that your body requires for normal function. So I did this with a fresh set of basket stars for 12 different animals. And this graph is a little intense, so bear with me. What I want you guys to take away from this is look at the level of oxygen as it goes across from left to right. And that's the dotted and dashed line. And you'll notice that when it dips down into the hypoxic level, the rate of respiration for the two specimens in black and gray inversely mirror that drop. And then they eventually recover in the post-hypoxic zone. So the takeaway from this was that, yes, the rate of bursal ventilation statistically significantly increases when oxygen is lowered. And all 12 of my basket star specimens did this. There was no variation, which was pretty cool to see. And then that cleared things up for the cold temperate water specimens, but I was interested in seeing what a basket star relative would do from a warm temperate region and from a tropical region. So I found a source of California brittle stars. They're the western spiny brittle stars, or Ophiurida spiculata. And I did the same oxygen reduction experiments on them. And unsurprisingly, they had the same reaction. The rate was higher for this smaller species, but every single individual that I worked on showed this increase in bursal ventilation rate when the oxygen dropped. And then my studies brought me to Panama with that GEO program and another Ophiuroid species. This one was the sponge-dwelling brittle star, and I had 6 specimens of that, and every single one showed that same increase. So this was more than just happenstance. This was definitely a trend. So that got me thinking, why would this weird behavior and this ability evolve and persist in not only cold temperate, but also warm temperate and warm tropical waters? So what was the connection between all this? And understanding, oceanography, all of these locations experience periodic seasonal hypoxia inundation. And basically that just means seasonally blobs of water move toward the continent and they have lower oxygen than the water that was previously there. And having the ability to regulate this respiration in temporary low oxygen events could have made the difference between surviving a low oxygen event and not surviving. And we see some support for this in the fossil record, which is super cool. Or we see historical evidence of hypoxia in the fossil record where brittle stars are present, which supports this theory. So in really, really low oxygen events, brittle stars will do this weird behavior called tipping. It's where they go up on their tippy toes, and then if the oxygen, low oxygen concentration persists, they'll flip over mouth side up and they will die that way. It's kind of sad to think about, but This has given us clues from fossils that show brittle star assemblages where all of them have died mouth side up. And then the chemistry from the adjacent sediment samples shows that there was also really low oxygen at the time that layer formed. So this orientation and chemistry suggests that brittle stars were in areas where hypoxia had been present for a long, long time. So if you are interested and want to read about this respiration behavior and the function of these internal brittle star organs in more detail, I encourage you to go check out our recent publication in the Biological Bulletin, and there is the DOI if you need it. You can also just find it by Googling Hainey and Emlet 2020. So I worked on some other projects while doing my master's degree, and one of those was figuring out the morphology and the anatomy responsible for this bursal ventilation behavior. And I did lots of drawings and descriptions of these anatomical parts. However, I really want to publish this year, so I'll have to keep my secrets for now, but I would love to talk about that at a post-publication date, so stay tuned for that information. But now I want to continue to talk about more fantastic brittle stars and why they're so cool, such as this jellyfish writing species called Ophiurida marmorata. This guy has a really cool life history where the little larval Pluteus forms will settle on these jellyfish, and they will filter feed and sometimes steal food from the jellyfish. And they grow and ride these jellyfish around the ocean until they either reach a large size or they sense the chemical conditions in the ocean that the sediment below the jellyfish is a great place to drop. And then once they reach this size or location, they will parachute off the jellyfish and live the rest of their lives as adults on the seafloor. So a really crazy but cool life history there. And some species of brittle star even exhibit bioluminescence. And bioluminescence is pretty common in the ocean. We're finding more and more species that exhibit bioluminescence. And I'm so happy that brittle stars do this as well. And we're not sure whether the main function is to startle predators or attract mates, but it could be both. So there are, in fact, representatives of this class that exhibit bioluminescence as well, which is just awesome. Some species have started to evolve rudimentary eyes, and they're not the classic eyes like you and I have, but their cells have started building these calcium carbonate lenses, if you will, and they are shaped just like the lenses in our eyes. And they have shown to bend light photons and focus them on excitatory nerve cells underneath those little bumps. And species like Ophiocoma wentai here in the right-hand corner react to movement overhead, so they can sense like light and shadow and movement, which can be very adaptive and advantage when it comes to evading overhead predators that may be searching for a brittle star snack. So the fact that these guys are evolving a different primitive eye is super cool. We talked a little bit about their ability to drop arms, which is a great predator evasion tactic, but it's not a super big deal because when they drop arms, they can just regrow them. And that's a really advantageous strategy, especially when dropping arms is one of the things you need to do to survive and evade predators. So here are a couple different photos on the screen of a brittle star arm with a regenerated length. It's the small spindly length denoted by RL. And then some photos on the right that I took of a basket star we collected. It was damaged in the trawl, but that new arm popped out very quickly after collection and then continued to grow several months down the road. And while I was putting this presentation together, I was doing some more research, and I found this really awesome paper about ophiuroids in human medicine. And this study done by Diaria et al. Was done in 2006, looked at 2 new compounds from Antarctic ophiuroids, and they tested them against various like viruses and bacteria. And they found that these 2 compounds from the brittle stars proved to be cytoprotective against the HIV-1 virus. And that's just a fancy way of saying that these 2 compounds protected human cells from the HIV-1 virus. So super cool study. And if you're interested, I've dropped the DOI on there so you can go see that. And it makes me think of the question, what other medical applications could brittle stars be holding on to that we haven't discovered yet? And then of course, finally, everybody's question is, where can I find brittle stars? And lucky for you, if you live near a coastline, your chances of finding a brittle star are really good. This is because they're everywhere. They are in every global ocean and sea. They can be found at every depth, from intertidal to deep sea, and they're in every climate, from the extreme poles all the way to the tropics. And if you live on the West Coast, like me, you can find them in tide pools. If you carefully lift up rocks, you might be able to see one trying to scoot back under the rock. And then what I like to do is go to the beach after big storms and find the kelp holdfasts. And I'll carefully pull apart the kelp holdfasts and look for brittle stars that are hiding in them, because they really like to hide in those nooks and crannies. And why they matter is always important to talk about, especially when they're an animal that's often not closely looked at. Not many people study their physiology and their role in the environment, but surprisingly, many things eat them. Everything from sea otters to crabs and fish. Here on the West Coast, brittle stars have been found in the gut contents of rockfish and halibut, skates, and Dungeness crabs. So they are a food source for things that us humans like to eat. So it wouldn't be a stretch to say they're economically important to us. But also, echinoderms in general, including brittle stars, produce 0.86 petagrams. That's almost 1 trillion kilograms of calcium carbonate per year. And calcium carbonate is super important for ocean water chemistry because it acts as a buffer. And we know ocean acidification is a problem that we are seeing effects of starting now, and we'll probably see in the future. So having this calcium carbonate buffer synthesized by these organisms is super, super important. And there are so many of these brittle stars all over the global oceans. They play a large part in that ocean chemistry. So that brings me to the end of my talk, and I hope you guys really enjoyed it and have come to appreciate them as much as I do. And on the screen here, I've left my website, my email, if you have any questions you want to ask later. And then if you'd like to stay in touch, I've left my public outreach sites on here. So my Instagram or my Twitter, where you can reach me and see what I'll be up to in the future. So anyway, thank you for joining me, and I'd be happy to try to answer your questions.
Great, that was amazing. There were so many cool things I was taking notes on for that, but we do have some questions. They were asked during the talk, so we'll do those first, but anyone else out there, you can raise your hand and I can unmute you to speak. I love doing that because it really feels like we're all together. You can also use the Q&A tab or the chat tab, whatever you would like. So the first question was from Brenda, and it was, how long does it take for a brittle star to go from embryo to adult?
Yeah, that's a great question. And it depends on a few things. It depends on species. So different species will have different development times. And it seems also that water temperature has an effect on how quickly those cells can organize and duplicate, and how quickly they can metamorphose from larval to adult form. So I don't have a definite answer for you, but it definitely varies.
Okay, and the second question, let's see, where did that go? Let's see. It's interesting that there are so many forms of reproduction in brittle stars. Does that help explain their long evolutionary life? Is it possible to determine the linear evolutionary progression of the different types of reproduction?
Yeah, it's very likely. In other organisms that have also been around for a long time, but also diversified, you see them filling different niches and utilizing different life history strategies to survive, and they do that successfully. So I think to answer your question, yes, brittle stars, their different modes of reproduction and development can probably be explained by the length of time they've been on Earth and how long they've had to evolve those different traits. So I hope that answers your question.
Sounds good. All right, Nancy is up next. Thank you, Nancy. I'm going to allow you to talk. There you go. Oh, it looks like. You were there for a minute.
I think you're still muted, Nancy.
I'm going to, there, how about that?
There you go.
Yep.
Well, McKenna, that was a really great talk, and you know, brittle stars they're really amazing. They're so diverse, and I loved hearing about all the different, especially the jellyfish writing one. How cool is that?
Yes.
So I have 3 questions, but I'll start with the one that's the most fascinating, which is that fossil record thing where they turn upside down and they're dead in those epoxy areas.
Wow. Yeah.
Are these linked in any way? For instance, you know, for instance, at the end of the Paleozoic, we had the big giant, you know, extinction, and then the asteroid hits and another one. So a couple of things. Are we able to link that to specific geologic events? And also, are we seeing more of it now because of climate change and some of these big epoxy areas we're seeing off the coast?
Yeah, so we probably could link them if the people who did these projects like carbon dated those substrates, but I don't know for sure.
Mm-hmm.
And then I was also interested in this hypoxia and their reaction to hypoxia because of the increasing frequency and duration of hypoxic events we're seeing on our coast.
Right.
But what we see now is that this regulatory ability that basket stars in particular for this area have evolved is not good enough to cope with the extreme hypoxia events we're seeing on our coast. So they will be negatively impacted by that, as you know, will so many other species.
So at the levels of oxygen that you tested, and they couldn't make it in some of these big sections where we've written like 0 oxygen for a month or 2 or even more.
Yeah, exactly. So they can't swim or crawl away quickly enough to get out of those zones. So most likely they will perish if they get caught up in one of those large, intense events again.
Cool. Well, thank you for that answer. That's interesting. And I was interested that the direct developers don't go through a larvae stage like the indirect. Like, I always figured direct developers just do metamorphosis inside the breeding chamber, but it looks like they even have a different way of developing.
Yeah, I'm not too sure about the details of direct developing inside the brood chambers, but I assume that the brooded ones do that direct development, but don't really know about details.
Oh, good point. They might brood and expel the larvae at different stages. You're right. Yeah.
That's true. Yeah. I just don't know those details.
Well, thanks for your questions, Nancy. We have so many more questions, so if you have a third one, maybe we can.
No, I actually miscounted. I had two.
Okay, great questions. Thanks, Nancy.
Thanks, Nancy.
Yeah.
Okay, so let's see here. Lori asks, how big does the giant green brittle star get?
I don't know what their maximum size is, but they are popular in the aquarium trade, and the ones I have seen about, I don't know, maybe a foot max. It's really interesting that they are in the aquarium trade, and novice aquarists often make the mistake of buying a bunch of them, thinking they'll be a great member of the cleanup crew, only to find their fish go missing at night. So they are large to be impressive in aquariums, but not so large that you can't keep them in a home aquarium. So I hope that answers your question.
All right, let's see. Jamie says, thinking about the ocean chemistry, do you know what percentage of that amount of calcium carbonate there is in the ocean? Sorry if you said this and I missed it. You might be able to see that too, McKenna.
I don't know the percentage, but we could probably figure it out if we, I think it was 0.86 petagrams of carbonate from echinoderms. If we got a updated number on what ocean chemistry is today, we could probably figure out that percentage, but I don't know it off the top of my head.
Okay. And Chip, I saw that you were raising your hand. If you want to, I can allow you to speak. Let's see, let me get. Chip, if you'd like to talk, you are unmuted. I've allowed you to talk.
Okay, I guess, yeah. So how similar are today's brittle stars to the ones that existed here? I think you said 500 million years ago, which is pretty mind-blowing.
Yeah. Morphologically speaking, they look pretty similar. It would be hard to tell what their internal structures look like. And personally, I haven't compared fossils with extant species, so I'm afraid I can't answer your question in detail, but just by looking at the two fossils and extant species, they look very similar. So few changes have happened, at least to the simple brittle stars and the early fossil species. So,
yeah, that's what I was amazed at. It was that they look very similar, and yes, of course, we don't know what they look like inside, but wow.
Yeah.
500 million years of the same kind of basic morphology. That's impressive.
Yeah, they found a good strategy. And
yeah.
stuff.
Thanks, McKenna.
Yeah, thanks.
Thanks, Chip. Yeah, when I was taking notes, it was just wow. I think I wrote wow like 5 times. A lot of wows tonight. All right, so Brent, who says he has spent a lot of time tide pooling, but he's never seen the brittle stars, so he's wondering what he's doing wrong. Thanks, Brett.
Yeah, so it probably depends where you are tide pooling. If you're on a really rocky, boulder-dominated place like Cape Arago, you might try going out as far as you can safely, and then flipping larger rocks that might have holes in that rock and looking within the rock itself for maybe an arm or two sticking out. But if you're on a more sandy intertidal beach, you might flip a rock and then maybe dig in the sand a little bit, and you can find maybe like the long-armed brittle star. We have both of those at several Cape Arago sites. So yeah, try to dig around a little bit. I'm sure you can find one eventually.
Thanks for your question. And I know that probably everyone who's here tonight will do a very responsible job of turning over a rock and digging on the beach, because we are all in the know about that kind of a thing. We know how to respect the beach. And so that's a great question. Okay, Dennis, it seems that you have confirmed that the brittle stars will wave their bursa to receive oxygen, but is it also not possible that they wave them for food without any oxygenation issues?
So your question makes me think that I should clarify that the bursae are internal structures. So they don't really wave those. They do wave their arms though. But let me go back to your question. Oops, it disappeared.
Here, there, it's in the answered, there at the bottom. If you go into answered in the Q&A and at the bottom and scroll down. Do you see it now?
Yeah, okay, there we go. Is it possible that they wave them for food without any oxygenation issues? So going forward with this question, they do wave their arms when food is present without any oxygenation issues. And then when they run around the tank, when their movement is increased, that it appears that that bursal ventilation movement also increases. So I think yes is the answer to your question, but I hope that clarifies and answers your question. Let me know if it doesn't.
Okay. Thanks, Dennis. All right, Susan asks, are brittle stars, particularly young ones, being affected by increasing ocean acidification? And then she adds a link and an article. Thank you, that's cool.
Yeah.
If so, have you seen any evidence of this during your research?
Yeah, so like you suggest, it seems that they might be struggling to acquire the carbonate ions needed to build their larval skeletons and the rudiments for the juvenile skeletons. And this is the case with all like shell builders. We see it with larval clams and pteropods. So it's not just the brittle stars that are being affected, but I didn't really work on ocean acidification, but that what I know in that article is basically what I know from reading other researchers' work. So yeah, that's what I know. And it appears that you are correct that they are being affected negatively.
Thanks for that. Also, everybody, I just put that link into the chat if anybody wants to check that out later. Lori asks, when oxygen level is stable, have you noticed an increase in the pumping motion when basket stars are feeding to when they are not feeding? I am wondering if the increased vascular activity also needs more oxygen or if the pumping helps the arm movement. Great question.
Yeah, great question. So this is kind of what my early feeding studies lended evidence to support. So when oxygen level remains the same and only food is added, this increased arm waving and increased bursal ventilation rate does occur. So this feeding and activity does seem to require more oxygen and potentially trigger this upregulation in bursal ventilation rate.
Okay. So looks like, let me look in the chat. Oh, Trish. Hi, Trish. Are brittle stars or basket stars being affected by sea star wasting at all? I realize they're not asteroids.
Yeah, thank you for bringing this up. I got asked this question a lot while at the CMLC. Trish, you probably still do. It appears that this disease does not affect Ophiuroids, the brittle stars and basket stars, like it's affecting sea stars or asteroids. They are, however, very sensitive to temperature increases and, of course, oxygen levels. we often see them start to fall apart and drop arms when the temperature increases, but it doesn't appear to be the same as sea star wasting syndrome.
I was very curious about that as well. And there's so much research coming out now about sea star wasting syndrome and the disease and what is causing it, that I encourage everyone to look for the newest news on that. It just seems to be changing all the time.
And I believe next week, Sarah Hamilton will be giving a public talk on the sunflower star and updates on sea star wasting syndrome.
Yes, exactly. And Shoreline for Education and Awareness is giving that talk, and I have links to that to register for that. And I'm sure that McKenna, so you can contact either one of us, and we'll put you in touch with C, who can then register you there. And I think that might be all of the questions. Thank you so much, McKenna. This was amazing. And I want to say too that McKenna is an amazing photographer, and almost all of the Brittle star and basket star photographs that you've seen tonight, if I'm not mistaken, right, were your photographs, right, McKenna?
Yeah, or they were otherwise accredited appropriately.
Right, exactly. I noticed that, but yours are the best. By far. They're just beautiful. And I'm wondering, is that a Brittle star on your shirt? It's pretty cool.
Yeah,
Did you make that?
I did make it. Yeah.
Oh, I love it. It's so cool. Well, now we all know what that is. It's just,
yeah. So I'm hoping now that you all appreciate Brittle stars more, we can also make them trend in fashion. So
that's exactly right. This was a wonderful talk, and we just had a comment that someone really loved the title of the talk, and I did too. And I encourage people to get to know their tide pools responsibly, know what's in there, and now that you've learned from this, and you can share the information with others that you meet down on the beach or in the tide pools.
Mhm.
Let's see, looks like we might have a couple more questions here. That was just a thank you. Oh, okay, one more question from Nancy, I think. Let's see. All right, Nancy, you are permitted to talk.
You're muted still.
Okay, I didn't know I had another question, but I actually do. What is the biggest brittle star?
Great question. Thanks.
Great
Nancy.
question. I don't know about brittle stars, but if you want to talk about Ophiuroids in general, I think that title would have to belong to one of the basket star species. Our local basket star, Gorgonocephalus, the really, really old ones that have intact arms can reach almost a meter across from arm tip to arm tip. And I think there are some tropical ones that could be contenders for the largest Ophiuroid. So they're very impressive animals.
Yeah, the basket stars look like they can just totally unwind and become very, very long. So I wondered the same myself. Okay.
One thing I wanted to ask is those ones that jump off the jellyfish, you said that they might be sensing a good habitat.
Yeah.
Wild.
Yeah, like a chemical cue. Not really sure. And the person who studied this wasn't quite sure either, but yeah, it's a very cool way of finding a good place to live that'd be suitable for the adult stage of that life cycle.
Well, it is 7 o'clock, and I like to keep these to about an hour because oftentimes people have another science webinar they would like to go to at 7 p.m. McKenna, thank you for taking the time with us and for creating this wonderful presentation. I've been waiting since last year when the Sharing the Coast conference had to be canceled because of the pandemic, and here we are about a year later. So it's wonderful to finally learn from you about these truly amazing creatures that I can't wait to share some of these facts with the Coast Watchers. I was going to quickly share my information as well, so that if anyone is here tonight that is not a Coast Watcher and you would like to be, or if you just have any questions about Coast Watch or Oregon Shores, go ahead and give me a call or send me an email. And here again is a fantastic photograph that McKenna took. And again, her website, which I highly recommend you check out. It's mckenna-marine-research.info. And thank you very much for being here. This will be available on the Coast Watch YouTube channel within the next few days. So you can share that with folks or watch it again with family or whatever you would like to do. Just contact myself or McKenna and we will give you that link. Thank you very much for being here. And thanks again, McKenna. It was great to see you.
Thank you. It was great to do this with you all.
Okay, good night. Thanks.